So in today's lab, we'll be looking at Koch's postulates. It's a 4-part lab, so we'll be doing 4 different sessions to go through the entire experiment. Just a bit of history. Robert Koch was one of the very important microbiologists who was present during the period of 1843 to 1910. He discovered many new methods of studying microbes and their relationship to disease. Robert Koch established the field of medical microbiology by his various inventions. Some of the important noteworthy things that he would have invented were solid culture mediums, plate culture methods, the use of pure cultures of isolates, or how to obtain pure cultures of isolates, as well as slide culture techniques. However, one of his most important and well-known discoveries would be the observation and experimental steps to prove that microbes are responsible for disease. He would have done this through studies of anthrax as well as tuberculosis. Today in microbiology, in the lab, we still use Koch's postulates to show the association of a microbe with a specific disease by looking at their relationship to the signs and symptoms that are present on different types of organisms, whether it's a plant or an animal. So for Koch postulates, we have 4 basic statements or tenets that are followed. The first being the diseased host is observed for signs of a pathogen as well as symptoms. The causal organism is seen to be associated with all diseased individuals or all individuals that show these common signs and symptoms present on them. The second postulate states that the causal organism can be isolated in pure culture and its morphology described. For this, we would be in the lab using a microscope to look at the morphological features of the organism. The third postulate states that pure cultures can be used to inoculate. Pure cultures of the isolate can be then used to inoculate a healthy host, and when incubated, that organism should show similar signs and symptoms as with our first postulate or our first sample of the diseased organism. And finally, the causal organism from this second sample can be re-isolated in pure culture, and again, when observed using a microscope, a light microscope, the morphological characteristics and features of this organism should be the same as our first organism that we would have isolated in the different stages of this lab. In the first part of our lab, we will be looking at understanding the application of the scientific method to Koch's postulates, and we will be isolating the pathogen and looking at its signs and symptoms present on the diseased organism. So here we have our specimen. For this lab, we will be looking at tomato disease. You can see clearly there are various signs and symptoms of disease present on this organism, this tomato fruit. So we are looking, the surface of the tomato has already been surface sterilized using distilled water. From this diseased organism, we are going to obtain an isolate, and we are going to do a quadrat shriek to try to ensure we get a pure culture that will be used in the second part of this lab. So as with following with aseptic techniques, the entire environment has already been swabbed with and wiped down, cleaned with 70% alcohol. We have our inoculating loop here, which we will be using to transfer a sample of the bacterial or fungal organism. We do not know its identity, but it will be used to transfer a sample of the organism onto a microscope slide. A gram stain will be performed on this sample, and the morphology of this sample will be recorded, as well as a quadrat shriek done in order for us to obtain a pure culture for our next session. So we have our diseased tomato here. We are going to take a sample from the region that you see signs and symptoms of the disease being present. We'll be taking a sample, we'll be placing it or doing a quadrat shriek for inoculation. We'll be inoculating a quadrat shriek for incubation to look at the organism after it has been incubated, as well as we'll be putting a sample onto a slide to do a gram stain on. So first thing we need to do is pierce the skin of the tomato to ensure that we're getting a proper sample of diseased tissue. So we're flaming our forceps. Make sure we practice all aseptic technique in this lab. to prevent contamination of our sample. So we flame until it's red hot. It's a little more difficult to see the red discoloration of our forceps compared to your inoculating loop. We immerse into alcohol and we flame off our alcohol. Remember, whenever we sanitize or sterilize equipment, we have to wait a couple seconds to ensure that the heat does not kill the organism that we are going to inoculate. So just remove up some surface of the tomato skin in the region where you see signs of the disease. So you have exposed there. Remember to again flame your forceps to remove any contamination. And then we're taking our inoculating loop, sterilize, and then we take our loop and we stick it in to the region of tissue where we have the organism present. We want to make sure we get a good sample there on our loop. And with that, we're going to do a quadrat shriek. So we have our nutrient agar plate here. And if you all remember how you're doing a quadrat shriek, we are streaking in each of the 4 quadrats of that plate. Now last streak, we bring it down to the center. Remember, you're not piercing your agar. After, we flame our loop. Again to re-sterilize. And our second step is for us to take a sample for our gram stain. So again, in the same area we would have taken our sample for our quadrat shriek, we are again collecting a sample of our diseased organism. And on a glass slide with some water, with a tiny droplet of water, we deposit our sample. Sterilize. And we'll have to heat fix this specimen before we complete our gram stain. So we could use our forceps there to hold our slide. Now remember, when you are heat fixing, you do not hold your sample directly over the flame. You can move it across the flame in intervals, and you're doing this so that you do not kill your specimen. And as you do that, you will see it's becoming fixed to the slide. you will see the water drying up and your sample becoming affixed to your slide. So remember, with your Bunsen you can manipulate your flame. So as you can see, the sample is almost entirely dry. You can leave it in your container, let it cool off a bit, and we're going to move across now to the sink to complete our gram stain. For our gram staining procedure, you will see we have all of our different requirements. We have our solutions, the first being crystal violet, grams iodine, decolorizer, and gram safranin. So in your lab handout on page 30, you can follow along to be reminded of what your gram staining procedure is. So you have your specimen here, has already been heat fixed to our slide. The first part, or the first stage of our gram staining procedure, is to apply crystal violet for 20 seconds and let it stay for 20 seconds. So we ensure the entire sample has been covered and we allow it to stain for 20 seconds. After our 20 seconds has passed, we wash off our crystal violet stain for about 2 seconds. You can just tilt our slide to ensure all of that stain has been removed. Next step, we apply grams iodine, a couple drops, 2 to 3 drops of grams iodine, and we're leaving it for 1 minute. So you start your timer for 1 minute. After 1 minute, the next step is step 4, decolorize with alcohol. So we have our decolorizer here. We hold our slide again at a tilted angle and we apply decolorizer. It's recommended about 10 to 20 seconds until your solvent flows colorlessly. So depending on your sample, you will know how to manipulate that 10 to 20 second time. So I have a colorless flow there. Then you wash with distilled water, 2 seconds. And then finally, our fourth solution, safranin. We add a couple drops of safranin, 2 to 3 drops of safranin, and you leave it for about 20 seconds. Finally, we wash out our safranin. Again, hold at an angle so it can wash off. And then we blot dry. Now we are not blotting the region where your sample is present, you are blotting around it. Your tissue will pull up the water from those regions of your slide. You want to make sure you get rid of any excess water because that is going to affect how you can see your sample under your microscope. So your gram stain has been completed. Now you're going to take your sample, put a cover slip on it, and look at it under your microscope to see what morphology or what morphological characteristics this organism is displaying. So we have placed our slide under our microscope. As with any specimen, when you're looking at a microscope, the first thing you have to look at it under is your 4 times magnification. So we have shifted our objective lens to the 4 times magnification. We're going to make sure we get a clear image by using our fine and coarse tuning knobs, as well as we can use shift our stage left to right, front to back, using our shifting it right or left to center it on our stage so that we can see our sample. We use our coarse tuning knob to get it into range, and then we move to our fine tuning to get a clear image. Once you have a clear image at your 4x objective lens, you can then rotate to your 10x magnification. This will allow you to see some more morphological characteristics of the organism as we increase our magnification. Again, using your fine-tuning knob, you get a clear image of the organism. And we can go up to our 40x magnification to get a final idea of what our specimen looks like. So some morphological characteristics to note would be whether or not your sample is gram positive or gram negative based on the color of the specimen after the gram staining procedure. Additionally, you are going to look at shape as well as arrangement of the organism. So this is what your slide, or this is what your sample would have looked like under your 40x magnification. So from this first part of your lab, according to Koch's postulates, we would have observed our diseased tomato. For signs and symptoms, make recordings of what signs, symptoms, discoloration of tissue, what type of lesion we see, if you see any substances oozing from the site of infection, and so on. So those things would be recorded as signs and symptoms of the disease being present. We would have further isolated our specimen and did a quadrant trick on a nutrient agar plate. Remember, you must label your plates up according to what information you are given for this lab. So this would be our Koch Postulates Lab 1. It's an unknown specimen. You date it so that you'll know that you're incubating it for what period of time, whether it's 48 hours, 72 hours, and so on. And you also put your name on it so you can identify it from those that your classmates would have done. And this will be inoculated for 3 days to look for growth of pure cultures or pure isolates, which are going to be used in part 2 of your lab. So that brings your first part of the lab to an end. You have your post-lab questions that follow in your lab handout that you must answer.